Hi. Good morning, students. Today we will be having a new lecture, which is in line with our discussions on cells. For our learning targets, at the end of the lesson, I can characterize the phases of the cell cycle and their control points. Realize the importance of cell cycle in the perpetuation of life by citing examples. Participate in class by sharing ideas and answering specific questions. Let us proceed to the students. Realizing the importance of cell cycle in the perpetuation of life. On the figure, you can see a butterfly that drink nectar from blossoms on flowers. It resembles life itself. Now students, perpetuation of life means continuation of species. This is ensured by reproduction. Since reproduction aims to increase the number of organisms of a species over time. In line with that, what if cell cycle does not exist? Can that species of butterfly continue to exist? So what is cell cycle to you? I want you to reflect on the importance of cell cycle. Today's topic focuses more on cell cycle. This is our 6th lecture in General Biology 1. So sit back, relax, and take down notes, students. Let us begin by defining cell division first. When we say cell division, it involves the distribution of identical genetic material or DNA to two daughter cells. What is most remarkable is the fidelity with which the DNA is passed along, without dilution or error, from one generation to the next. Cell division functions in reproduction, growth, and repair. In line with cell division process that produces two daughter cells that has an identical genetic material. Cell cycle means that it is an ordered sequence of events that extends from the time a cell is first formed, from a dividing parent cell, until its own division. One of the distinct characteristics of living things is being able to preserve themselves. Cells need to undergo cycles as part of their growth and to repair or replace damaged parts. Cell cycle enables a living thing to continue its existence by multiplying itself in controlled and systematic processes. This lesson will enhance your understanding on cell cycle. This will provide learners with the concepts on the different stages of cell cycle, and their control points, and their significance on an organism. Now let us proceed with cell cycle checkpoints. It is the critical point in the cell cycle, where stop and go ahead signals can regulate the cell cycle. Animal cells have built-in stop signals that halt the cell cycles, and checkpoints until overridden by go-ahead signals. The three major checkpoints includes G1, G2, and M phase, which these three processes is in the interphase of cell cycle. When we say checkpoint, it means the restriction point. Before proceeding to the next phase, it should be halted, be checked, and be ready first before it may move to the next bigger activity. This is done in the G1 checkpoint. That ensures the cell is large enough to divide, and that enough nutrients are available to resulting daughter cells. If a cell receives a go-ahead signal at the G1 checkpoint, it will usually continue with the cell cycle. If not, it will exit the cycle and switch to G0. Now what is G0 phase? Multinucleated muscle cells that do not undergo cytokinesis are also often considered to be in the G0 stage. On occasion, a distinction in terms is made between a G0 cell and a quiescent cell, example heart muscle cells and neurons, which will never enter the G1 phase, whereas other G0 cells may. Cells enter the G0 phase from a cell cycle checkpoint in the G1 phase, such as the restriction point, animal cells, or the start point, yeast. This usually occurs in response to a lack of growth factors or nutrients. During the G0 phase, the cell cycle machinery is dismantled, and cyclins and cyclin-dependent kinases disappear. Cells then remain in the G0 phase until there is a reason for them to divide. Some cell types in mature organisms, such as parenchymal cells of the liver and kidney, enter the G0 phase semi-permanently, and can be induced to begin dividing again only under very specific circumstances. Other types of cells, such as epithelial cells, continue to divide throughout an organism's life and rarely enter G0. It ensures that the DNA replication in S phase has been successfully completed. Now in the M phase, which is termed to be mitosis checkpoint, that happens specifically in metaphase. That is why it is termed to be metaphase checkpoint also. It ensures that all chromosomes are attached to the mitotic spindle by a kinetochore. With regards to the chromosomes, the activity of chromosomes ensuring cell cycle is that the DNA containing cell's genetic code. Each chromosome has a matching pair, which is homologous in nature, meaning it has a specific pairing activity. As you can see on the figure, the M phase, which is the cell division, is just a small portion in the entire cell cycle. But this is a critical spot for it geared towards the end. That means the resulting factor of the cell cycle. On that part of the cell division, it involves 4 specific processes. That includes prophase, metaphase, which is where the checkpoint in the cell division, anaphase, telophase up to cytokinesis. On the figure again is the M phase, the cell division, comprising mitosis, when a fully grown cell segregates the replicated chromosomes to opposite ends of a molecular scaffold, termed the spindle, and cytokinesis, when the cell cleaves between the separated chromosomes to produce 2 daughter cells. In general, each daughter cell receives a complement of genetic material and organelles identical to that of the parent cell. Let us proceed with the kinase. It is a protein which activates or deactivates another protein by phosphorylating them. Protein kinases, PTKs, are enzymes that regulate the biological activity of proteins by phosphorylation of specific amino acids with ADP as the source of phosphate, thereby inducing a conformational change from an inactive to an active form of the protein. Kinases give the go-ahead signals at the G1 and G2 checkpoints. But, to start this, they must be activated. The activating molecule cyclin, a protein that derives from cyclically fluctuating concentration in the cell. Kinases are called cyclin-dependent kinases, CDK. Cyclins accumulate during the interphase. By the G2 checkpoint, enough cyclin is available to form MPF complexes or maturation promoting factor, which is the aggregate of CDK and cyclin. The MPF complexes initiates the mitosis by phosphorylating key proteins in the mitotic sequence. Later in mitosis, MPF switches off by initiated a process which leads to the destruction of cyclin CDK. The non-cyclin part of MPF persists in the cell as an inactive form until it associates with new cyclin molecules created during the next round of cell cycle. That ends our discussion on cell cycle. With regards to the Moodle, Module 7 is now available. Please check your Moodle account. Thank you for listening. God bless us all.